Hi, today I'm going to walk you through a lab. It's called DNA, What's It Look Like, provided by Kansas Corn STEM. Hi, I'm Bill Welch. I'm a master teacher at Kansas Corn STEM. Today I'm going to walk you through the DNA, What's It Look Like lab, provided by Kansas Corn STEM. Now, purpose of an activity like this, and based on the title of what we're looking at, what does DNA look like? DNA is microscopic. We have to actually usually make models to actually see what it looks like. So when we're looking at the objectives of this lab, it's important for students to understand how the small world operates at their ability to have a concept of some of these molecules they can't really see. So it's important for students to understand what DNA is and how all cells utilize it. DNA is a major structure of biological molecule in biology and living things. One of the major differences in a DNA lab is that we attempt to extract DNA from material. It could be strawberries, it could be human cells, and we get a small amount of that material, but that doesn't tell us the structure, shape of that DNA, okay? So this lab we're going to look at has three parts. And the first part, we're going to try to extract some DNA from canned corn. The second part of that is we're going to take a DNA modeling activity. We're going to model what DNA looks like. And then the third part, we're going to be student experimenting with DNA extraction procedure and kind of a guided, open-ended part to this lab. So let's start by looking at DNA. And again, when you've got your textbooks, you've got a lot of different structural diagrams you can look at. The diagram here is a very common diagram you'll find out in most life science and biology books. You actually can see DNA in the structure of chromosomes, but those chromosomes are very condensed DNA. And DNA is essentially wrapped around these structures called nucleosomes, and those nucleosomes are essentially histone proteins that are, all of this gets pushed together and tightly packed, and then when it's tightly packed, it actually shows, when you can actually stain it, you can actually see it as a chromosome, okay? So, the extraction part, we're going to do part one right now, and we're just going to extract DNA from canned corn. So obviously you're going to need some canned corn, okay? Ideally, the canned corn you need is, nowadays, not too many people have can openers. I usually get the ones that definitely have the lid open with it. It doesn't have to be any specific kind of corn, but any kind of canned corn is going to work, all right? The whole idea here is you take 100 mLs of just the corn, okay, which is pretty much half a cup of this canned corn. You're going to take a tablespoon of table salt, which we've got right here, okay? So you'll have a tablespoon of salt, and then you're going to add 200 mLs of cold water. And you're going to put all this in the blender. And it's going to look like this, okay? The major emphasis here is trying not to mix it all up too quickly. So when we blend this together, you're going to have a big, and you're going to let it run for maybe 30 seconds to a minute. Once it's finished running, okay, you'll have a mash. And that mash will, this will be a whole, we'll just say a big gloop of stuff. And some of the stuff what you're going to need to do now, and this is going to be the next step we're going to go to, is you're going to pour this through a strainer. Here's a strainer, and you're going to pour that into a cup, okay? Now, make sure that this, again, once you pour this material, this mixture in here, some of the solids will get stuck in the little basket, and then some of the liquid will drip through. We want the liquid through there, so we're not going to try to pour this through there fast because we don't want stuff going over the outside edge. We want the liquid portion of this extraction material to come through there, okay? So that's step one. And then what we're going to do in this container, this structure, this solution here, we're going to slowly add roughly 2 tablespoons of liquid detergent. And this, ideally, a non-colored liquid detergent works better. And when you add this, the whole idea there is that you're adding it slowly so it doesn't, if you just throw it in there, you're going to have a bunch of bubbles. And those bubbles are not going to help us, okay? So once you get your 2 mLs of soap in this solution, you can actually let this sit Because what we've got in there, now think about it, we've got salt, we've got soap, and then we've got this mixture of corn and water material. Proteins, DNA, nucleic acid, all this other stuff that's in there, carbohydrates, everything that's in corn is still in there, okay? Except for the solids that we've actually filtered out, okay? So, let this sit. We've got a salt that's going to be doing some work in there. We've got a detergent in there that's also going to be doing some work, okay? Once we've got that, this is sit. We do not want to make this move to where there's bubbles in it. Once we've got that set, we actually can pour that into a test tube, okay? And for time purposes, I'm just going to show you what that's going to look like. Once you pour that in, and the test tubes you're going to get are these plastic test tubes, and it says pour about up to 1/3. Obviously, I over poured this one, but the whole concept here is that you've got this material that's in there. And think about what we have in there. It's very simple. You've got liquid corn material that came through. You've got salt, and you've also got liquid detergent. Okay, all of that's together in there. And now, what we're going to do. Now let's take a look at what the purpose of this material is. This salt has a purpose. Those histone proteins that we had in there, the salt's going to pull those proteins away from the DNA. Okay, the liquid detergent, it's going to open up the cells so that we can get to the DNA. Okay, so all that's in there, and that's all working inside that test tube. And then the next thing you're going to do is you're going to add a little pinch of meat tenderizer. We're going to use meat tenderizer as an enzyme. Okay, so a little pinch. Okay, that little pinch does not have to be much. This meat tenderizer container should last for a long time in large classes. So you're going to take a pinch of those enzymes. You're going to put that in there, and once you get those enzymes in there, you're going to, and by the way, the enzymes are another part of trying to open up the cells where the DNA is, okay? So, once that's done, the last step will be to take some ice cold, and you can take ethyl alcohol, isopropyl alcohol, either one will work. Ethyl alcohol seems to be the best. It usually is a little more pure. And what you're going to do is you're going to slowly add this to this mixture, okay? So, if you're looking at how that's going to work, it's very simple, okay? When you tilt this to the side and you slowly add this alcohol, and it has to be cold, because if it's not cold, if it's room temperature, it doesn't get down into the mixture like it's supposed to. So, for instance, when I'm going to add this, and there's already some in there, but I'm going to show you how I'm going to add this very quickly. You're going to just gently let it run down the side of the test tube, okay? You don't have to do it fast, and the whole goal is not to get finished first. It's to get it in there, and once you've got your volume in there, okay? I'm going to put that much in there. Once that's in there, then you are going to let this sit. Now, again, you don't want to shake this up. You want this to sit, and a test tube rack would be good. A little cup is just as fine. All you need to do is make sure that it's going to sit there and not move, okay? Because right now, where this cold alcohol is, that layer is going to hit where all these proteins and nucleic acids and carbohydrates are sitting, and it just so happens that that DNA is going to be right at that layer where that cold alcohol is hitting that other material. That's where that DNA is going to precipitate out. And what's going to happen is it's going to be pulled up into that alcohol layer. So when we look at this, in this slide, you're actually seeing some of it start to come out of that bottom area. Now, again, depending upon how, if we mix it up good enough, do we add the right materials in the right order, this might take 4 to 5 minutes to occur. It's not going to be instant, okay? A lot of times you do DNA labs with something like strawberries. Strawberries they've got 8 times the amount of DNA that something like corn does, okay? So let's look at this and wait, let that sit for 4 or 5 minutes, and then you're ready to move on, okay? And that'll be the end of part 1. Okay, so for part 2, we're going to try to make a model of DNA and what it looks like so we can visualize it at a macroscopic level, not a microscopic level, okay? So to do that, in this kit you get, you're going to have some little they're ping pong balls and these little Velcro tabs. And what you're going to need to do is, when you give these to the students, you're going to have them, each group should be able to make one of these little 4x4, it's actually 2x2. This is a nucleosome. It's going to take a total of 8 balls to make that, eight of the ping pong balls. And you've got the little Velcro tabs, stick tabs on there. Believe it or not, this takes a long time for some kids to figure out how to do this. You need the opposite Velcros, you need to be able to stick it together. Ideally, it sticks together pretty solidly. And now I've got, in this case, if you look at this, I've got 4 nucleosomes. These are the proteins that are going to, that the DNA is going to be wrapped around. Why does DNA have to be wrapped around it? This is actually what helps protect DNA from breaking apart, okay? So, in this case, when we're looking at that, the next thing you need to do, so each group's made a nucleosome. Now you're going to say, we're now going to try to add the DNA to this nucleosome. To do that, all we've got to do is get some DNA. And what we're going to use for DNA is very simple. Believe it or not, DNA is almost completely clear. We have plastic wrap that we're going to use. And again, the procedure for this, it depends, especially if you've got 6 lab groups or 8 lab groups. I would probably get you 8 to 10 feet of this. It doesn't have to measure out perfectly. And again, it's going to look something like this. And what you're going to do is have each group come up one at a time. And this is essentially what you're going to look like. It's going to be one wrapped nucleosome. Okay? You wrap it across the other nucleosomes. Leave a little space. Okay? And as you do this, you'll have, assuming I've got enough of this DNA that I've pulled out of that saran wrap container, I now have DNA wrapped around these histone proteins, okay? So this is actually an uncondensed version, okay? Don't forget, this is the DNA, this is the proteins, okay? So these are the proteins. Now, when we push these together tightly like this, okay, when these are tight together, that's a chromosome. That's in the, for instance, if you were getting a karyotype, this would be at the the metaphase stage where they'd take a picture of it. So we've got a condensed chromosome. And the chromosome themselves are found where in the cell? Well, they're found in the nucleus. So we've got to have a nucleus, okay? So we pull up, we'll use this little clear bag as a nucleus, okay? So we can take our chromosome. So the class, we've got this chromosome that now is inside this nucleus. You could tie this off if you wanted to. For our purposes here, we've just got a nucleus with some chromosomes in there. And a chromosome, which is made up of histone proteins, the nucleosome, and also the DNA. And this is the nucleus, and the nucleus is only a cell organelle, so in the cell, it's going to be one part of the cell. So here's a bigger sac, theoretically. Now I've got the nucleus inside the cell. So if you look at this, we've got the cell membrane, we've got the nucleus, the nuclear membrane, and inside that we've got the DNA that we're looking for, okay? So when we try to extract DNA from cells, corn in this case, we've got to get through this cell membrane, which is made up for the most part, it's bilipid, lipid is a fat, okay? And then the next membrane is the nuclear membrane, okay? That's also a bilipid layer made up of fats, okay? So we have to get through 2 fat layers, and then once we get into the 2 fat layers, the next thing we have to do to get to the DNA is we need to actually get the DNA separated from these nucleosomes or histone proteins. So somehow we're going to need to get these things pulled away from the nucleosome. That's good Velcro. Okay. And this one didn't even go through. It went through this way. And look at this. Now we've got proteins. These are the nucleosomes. This is a protein. This is the DNA part of the cell. Okay. This is what we're trying to get. Okay. So once we've get this, when we have this, and if it's purified and we've got it correct, we can actually do something with this DNA. Okay, that's the universal code for everything, for all living things. Now, to actually do this, we physically, and if you look back at what we're trying to do here, okay, we added salt. Salt is a material that will precipitate out the proteins. These are the proteins. The proteins that are part of that we're holding the DNA together, okay? The actual soap that we used, okay, was essentially, and soap, I'm going to show you here real quick with a quick demo, what happens when we break down a fat, okay? Lipids, which are fats, make up the membranes of the cell membrane and the nuclear membrane. When we do that, we are essentially using something to break apart the fat molecules. And to do that, we use detergents. It's a surfactant, okay? So this soap is to help open up the membranes that they're, this corn has a bunch of cells in there. We want to open up the plant cells, cell membrane. We want to open up the nuclear membrane. And this soap is what's doing that, okay? Now, just to show you this, okay, for instance, we've got another example of a fat, lipid. Butter is an example, okay? And we're going to stop here real quick. I'm going to heat this up and show you what this looks like, okay? So I melted the butter. This is going to represent a cell membrane, plasma membrane, and also nuclear membrane, okay? It's made up of fats. What? Because I told you that we're trying to break up the membranes by using a surfactant, a detergent. Well, watch what happens when I put a little drop of this detergent inside that. So I put a drop in there, and you notice, if you can tell, it's wherever the drop hits, this fat moved away from it, okay? That fat moved away from it because it's essentially moving in between the molecules of the fat molecules. So it breaks it apart. So that's how the membrane is getting broken up, by using a detergent, okay? That's one example of the material that we're using. Now, the other example I've got here for you, it may not be the best example of how are we going to get rid of this material, these proteins in there. Because if you've ever extracted DNA from a lot of different material like strawberries, you see a lot of this stringy white stuff. Well, I just told you this is DNA, these are proteins. So if you got stringy DNA, you've got DNA with these proteins in it still, right? Okay? So the whole idea, if you're doing this in an actual research lab, they just want this with no proteins. So they use specific salts and salt combinations. But this actually has milk. Milk essentially has albumin, which is a protein, okay? And when you add salt to milk, and it's going to be hard for you to see this, I'm going to add some salt in here and move this around a little. And what happens here over a fairly short period of time is you will see a separation of liquid portion of the milk, and there's going to be a part down here at the bottom that looks like something's falling to the bottom. And you're saying, well, that's just the salt. Well, it's not just the salt. What happens is it's the salt and any of the proteins that it actually is coming in contact with. So you're essentially pulling out the proteins in that case, as far as that goes. So this is just a quick separation, giving you a little demonstration of how to separate, don't forget what our goal is. We want to look at the structure and find out how what DNA looks like. So we created a little model of it. I tried to give you a couple demonstrations about how that model correlates to these materials that we use to try to help separate, okay? Last thing we want to try to do here before we get to our last step is to take a look and see if we can see any precipitated or some DNA. Now I told you the DNA is supposed to be right here at the surface, the interface between the clear, the cold alcohol and the material, whatever else is there, okay? So ideally, this is where you're going to see some of this material. What you're going to probably see are little white strands in there which means they still have some protein in them. And I don't know if you can tell, there's a little cloud of whiteness down there. If you took it could be the glass string rods work sometimes. I found, to be honest with you, that a dropper pipette, if you go in with a dropper pipette, you can actually pull some of that material up just slowly. And once you get it out, you'll find out that it's not the white material, it's actually a clearish material. And that clearish material, I say clearish, it's not a color, but it's actually some of the DNA. And that clear DNA is really what we're trying to look at. Now, based on just what I did right here with this quick setup, I don't see a whole lot of DNA there. So the challenge is going to be what our next step is. Our next step is going to be right here. Okay, how do we get more DNA? Okay, so I've got corn. I've got all these materials here for students. And I actually, I think one of the best things you can do for students, and when you're talking about in science, is have them try to figure some of this out on their own, okay? So this is more of a guided, open-ended approach, and this is, every group will have this project goal. You're gonna try to obtain the greatest quantity of DNA from 3 tablespoons. Each group will just get 3 tablespoons of corn, and you can only alter the materials we provided. You can't get anything extra, you can't do anything. We're only altering the materials, the quantities of the materials we used. Okay, you can either use the materials or not use the materials, but you can't bring anything else in, okay, as far as that goes. And that opens up the thing, is the idea of, yeah, because this, you're gonna get some complaints, hey, there's not a whole lot of DNA there. And I'll say, yeah, I know that. Try to figure out how to make more DNA there, or obtain more DNA there, okay? So a very common whiteboarding activity is a CER, claim, evidence, and reasoning idea. And this whiteboard activity is pretty simple. You put in the question, the research question, okay? And this is an example right here on the slide showing you how that can be done. Let me give you, I don't know if this is the best view for you, but what we're looking at here is how can you get more DNA, the most DNA out of the cell and then the nucleus. And the claim is the students could say the DNA can be removed from the cell by adding more salt, adding more enzyme, putting more detergent in there, or blending it longer, or all these different characteristics they can do. They're going to actually try to make a claim, and then they're going to go present this, go ahead and collect the data, find out what they get, look at their results, and then based on that, they'll have some evidence. Once they have their evidence, then they can make their claim, their reasoning, sorry. Their reasoning is going to be very simple. Oh, we did this, we changed the amount of salt, and we didn't get any more DNA. That's not a bad lab activity. That's actually an experiment that tells you that, no, that didn't work. That just means they've got other options to look at, okay? And that's really what we're looking at with trying to get some open-ended work with claim, evidence, and reasoning. And again, it gives kids an opportunity. Now, how much time you want to spend on that is up to you, but that part of the activity is where they get to actually do some open-ended thinking. And other than that, they're just passively looking at and listening to you. That open-ended thinking is where they really learn and retain it. And that will be the what does DNA look like lab activity.